Hi everyone, welcome to part 2 of the lab design video. In this part, we are going to focus on the experimental setup. Let's get started. We start the experimental setup with the conditions of the control. Now, in every experiment, your control is going to be the baseline you're going to compare all other experimental setups to. So your control serves as a way to compare whether or not something got better or worse in your experiment. Typically, there will be two types of controls. A negative control is a setup where no reaction is expected. So for example, if you were testing an enzyme, you may have a setup that has just plain water and no enzyme added. So you would expect no reaction to take place. A positive control is a reaction that is expected for normal conditions. For example, an enzyme at normal temperature. So for example, if you've ever gone in for allergy testing, what they do is they do the allergy test on your skin. And what they do is they prick you just below the surface of the skin with various known allergens in your area. Now, when you look at the setup of the allergy testing, every single one of these is a different allergen. So this could be cedar, this could be milk, this could be mold, pet dander, ragweed. All of these could be different items. But within this test, within this allergy test, you're going to have a positive and negative control. So the negative control, you get pricked with just plain water. This is not supposed to produce a reaction, and they want to make sure that it isn't the needles, the tiny needles, that are causing you to have a reaction. So this is a negative control. It's plain water. The other one is a positive control, and so this is going to be a chemical called histamine. This is what causes the itchiness, the redness, the swelling. This is known as your positive control. Then what they do is they go ahead and test all of these different factors, and they let it sit for a while, and then they come back to check to see, do you have any welts? Are there any red spots? Whatever you react to that matches that positive control means you're allergic to it. Next, we're going to talk about factors that remain constant. It's important that in every experimental you set up, everything remains constant except for the independent and dependent variable. By doing this, you can help to ensure that the experiment is going to be fair. So everything except the dependent and independent variable have to be kept the same. For example, let's say you're doing an experiment where you want to test to see the effects of fertilizer on the flowering of plants. So you want to see how well the plants flower when fertilizer is added. Now think about the impact it will have if there are not constants in this experiment. So in other words, if A gets more water than B, then when you add the fertilizer, let's say we're adding the fertilizer to B. If A is getting more water and grows better, then that's not a fair comparison. Likewise, let's say that pot B is getting more sunlight compared to A. Well, then how do you know that it was the fertilizer that made this plant better? So in your experimental setup, you've got to make sure that everything stays the same between your experiment. So we're going to add the fertilizer to here because it's our independent variable. But things like sunlight needs to stay the same between both plants. The amount of water needs to stay the same between both plants. Maybe the size of the container. All of these variables need to stay the same between the two experiments so that we can make sure that it's just the addition of the fertilizer that's going to impact the color or the amount of flowers we get. The last thing I want to talk about are multiple trials to confirm your results. Typically, if you want your data, if you want to better understand your data, you need to make sure you do your experiment more than one time or on more than one subject. An easy way to take care of multiple trials is to set up an experiment where you're calculating an average. This way, if any one of the experiment fails, you'll still be able to get data from the rest of the trials. Another way to do this is another way to set up your multiple trials is to have a high number count. So for example, if I was taking a survey on the choices that students make at lunch, asking 400 students will get me much more understanding and reliable information than asking 10 students. Thank you all so much for watching. I hope this video was helpful. Bye.